I think it's probably one of the most cruel illnesses that that I know of, certainly. And when you see a young life, or a child developing until he's about 8 or 9, and then losing all the abilities that he's had, and then dying when he's only in his early teens, I think that in itself is a very powerful reason for wanting to continue research in whatever way that can be allowed by society. And I think the society needs to be told of the ways that people with a handicap and an illness like our boys had can be helped by science.
We are on the brink, I feel, of breaking through in a fairly big way in research that could help overcome genetic diseases, and that it would be totally irresponsible, to my view, to prevent this sort of research from going forward, as would be the effects of the Powell Bill.
The nature of the illness was that when they were born, they appeared normal, whatever normal is. And they were very happy children, always full of love and very, very happy in the home. They developed slowly, but they did progress. Everything was slower. I mean, language didn't come at all very well. The toilet training never really arrived. The walking and the mobility was a problem generally. Until they were 8, I think that they progressed. Mm-hmm. And then after that, there were signs that they were not progressing and they were really getting back. But it was the awful thing was then seeing them generally deteriorating and feeling as parents the terrible, terrible agony of not being able to do anything to avoid this.
Inherited genetic diseases put a great strain on the unfortunate sufferers and their whole family. The effects of some diseases can be quite devastating.
Well, if you take Duchenne muscular dystrophy, which is perhaps one of the most tragic conditions, really, because the child, and it's a boy always, is born apparently entirely normally. There may be some hints that they are a little bit slow in a proportion of them in their development, particularly in their walking. They may not be walking at 18 months, and not that one would be greatly alarmed, but one's beginning to think, is there a reason for this? Then, over the next year or so, they'll see a boy who's been running around perhaps gradually find it less and less easy to get up from a sitting position, has to sort of climb up his body, as it were, you know, with the support of his hands in order to stand up. That boy will gradually go into a wheelchair by the age of about 11, and at the present time, most of them will die by the age of 20.
Surprisingly, genetic abnormalities are present in about one family in 2,000, and they often have the same relatively simple general causes.
We can be very certain now that the whole difference between somebody who has Duchenne muscular dystrophy and somebody who doesn't resides in a change in the genetic code for one gene or a class of genes at a particular point on the X chromosome.
Genes carry the genetic codes for a population. They can exist in different forms called alleles, which have different effects. There can be faults in the alleles, and often the faults emerge quite unexpectedly.
We had no indication whatsoever in either of our two families, in the previous generations that we know about, that there had been any sort of disability like this whatsoever. It was a total fluke that there was a coming together of genes that in fact did have the possibility of leading to disablement. And in that way, this can happen to anyone.
The parents could have a normal allele and an abnormal allele. They themselves would be perfectly okay if the normal allele is dominant. The danger comes when both parents have the defective allele. The chance is that on average 3 out of 4 of their children will be normal, as they will inherit at least one normal allele. But there is a 1 in 4 chance of a child inheriting 2 abnormal alleles and so developing the disease. Although the other children will not have the disease, 2 of them have the defective allele, and may pass it on to their children. In recent years, some medical scientists in places like the John Radcliffe Hospital in Oxford have been trying to study these genetic diseases at the biochemical level.
It's mutations at the DNA sequence level. That means you get changes of the DNA sequence which alter the protein product that is being produced, or actually stop the protein product from being produced. Well, it can happen at all stages, either DNA being transcribed into RNA, or it can be a mistake in the RNA being translated into the protein, where you can actually get malfunctioning protein made instead of the normal protein being made.
The DNA is made up of a sugar phosphate backbone with different bases attached to it: thymine, adenosine, cytosine, and guanine, arranged in different orders. It's that order which determines the genetic code, the code for making proteins, such as enzymes and so on. The DNA strands can unzip and act as a template for the formation of a substance called messenger RNA. The order of the bases in the messenger RNA is determined by the order of the bases on the DNA. The DNA is a code, and so the RNA carries away a coded message. Different parts of the biochemical processes occur in different parts of the cell. For instance, when the messenger RNA molecules have been synthesized, they leave the nucleus through the nuclear pores. Once outside, the RNA is transported to the ribosomes on the rough endoplasmic reticulum. The ribosomes are made up of proteins and another type of RNA called ribosomal RNA. The messenger RNA lies across the ribosomes and acts as a template for incoming transfer RNA units. The transfer RNA joins up in an order determined by the order of the bases on the messenger RNA. The transfer RNA has an amino acid attached to the other end. The result is that amino acids are brought close together and join up to form a protein. That's how proteins are synthesized. The exact protein formed, the order of the amino acids, depends on the order of the bases in the messenger RNA, which was determined by the order on the DNA. So if there is even a single fault on the DNA, it will produce a wrong base on the messenger RNA. In turn, a fault in the messenger RNA will result in the wrong amino acid getting into the chain. Even a single fault in the DNA could result in a completely unsuitable protein being produced on the ribosome. As scientists learn more about this biochemistry, they are more able to give better advice to anxious couples.
If somebody is anxious, they said their uncle died of this, or their sister had died of some disease, and they didn't really know what it was, and they were concerned about risks for their own children, then genetic counselling clinics are very happy to see them and to sort it out. And so often we give the good news. They come fearing they've got some awful thing in the family, and we can reassure them.
We become involved in particular with X-linked diseases in the case of Duchenne muscular dystrophy, for example, when a lady will present at the clinic, at the genetic counselling clinic, asking for advice because there's Duchenne muscular dystrophy in her family, or she's already had a Duchenne muscular dystrophy boy. Well, first of all, they'll send us blood samples from all of the family members that they can collect. The affected boy, any normal brothers of the mother, and so on, so that we have the complete family so that we can look at the genetic inheritance of the probes we look at with Duchenne muscular dystrophy. It consists of taking out the long DNA strand from the cell, from the blood cells, and then cutting that up with restriction enzymes into little pieces, and then looking at the sizes of those little pieces in a defined way with a radioactive probe.
The scientists are looking for the way the defective alleles are handed on. They take samples of white blood cells from all the family. They put a radioactive marker on the DNA near where they think the abnormal allele is, and then break up the DNA with special enzymes.
Well, this is an example. We've got here this is a control on the end. Well, this is the normal control pattern. This is the affected boy with Duchenne muscular dystrophy with a lower band. This is his sister with 2 bands, and you can see that her father had the upper band pattern, and her mother has a 2-band pattern. She's therefore inherited this top band pattern from her father and this bottom band pattern from her mother, which is the same as the affected boy, which means that she is at high risk of being a carrier for Duchenne muscular dystrophy.
We can then warn them that in any subsequent pregnancy, there is that 1 in 4 chance that the child will get a double dose again. And then we can start talking about what can be done, the options and so on.
It's not practically possible to test every person in the country, but obviously high-risk couples don't want to bring an abnormal baby into the world. Luckily, there is now a test available during early pregnancy. A small sample of the fluid which surrounds the baby is removed and tested. To avoid damage to the developing embryo, this test can only be done at 18 weeks of pregnancy.
That is really very late in the pregnancy. The baby is already moving, and the mother's become very attached to this little thing that is inside her. So it was really with very great excitement that we realized that you could in fact get tissue from the developing embryo very much early on in pregnancy that could be used for prenatal diagnosis.
But why is that early test so much better?
Firstly, of course, that if they do have to face a termination of the pregnancy, it's very much earlier, and it's much less stressful and medically much safer. But I think there's another very important reason, and that is that it's early enough in the pregnancy for the couple alone for the pregnancy to be a secret between the couple, really. They don't have to tell anybody about it.
These are private decisions for the couple themselves, decisions with enormous consequences.
But we did go on for our 4th child, knowing what the situation was, and full knowledge. But with the sort of safety trap, I suppose you could call it, that we could face or could be helped with a termination should he be affected. And at the time, we neither of us had any doubt that and even more now, that we wouldn't want to bring another life into the world to have the same sort of illness and disability the eldest 2 had.
For the 1 in 2,000 couples who find themselves in this dreadful position, the main desire would probably be just to start a pregnancy knowing that the baby is normal. And now, at last, there is a chance to help them with the development of existing techniques. Scientists think eggs and sperm might be removed and fertilized in vitro. The fertilized eggs would be allowed to divide several times. Then small samples might be removed. At this stage, the eggs themselves would be frozen. The sample cells would be multiplied and tested. A normal egg with no abnormalities could then be identified and only that one replaced in the womb.
And so they get round, the Russian roulette really is brought right forward to choosing them in a test tube as opposed to killing them off in early pregnancy.
The recent rows in Parliament were caused by the suggestion that the necessary research might be done on the so-called spare embryos, which are byproducts of test tube fertilizations.
There are a number of spare embryos, which in practice at the moment are washed down the sink. That's the reality. And what we're saying is that rather than have a useless existence for a few days, those clusters of cells could be of enormous benefit to medical science in being observed for a period of time to see how they react to certain circumstances, differences in temperature, differences in nutrition, differences in the circumstances in which they exist. And that may give leads to overcoming a disability.
That technique would take away the element of chance. Other work depends on first finding the abnormal gene, and that's no easy task.
We're within 1,000 kilobase pairs of DNA sequence, I'm sure, and we and other groups are working very hard now to try and narrow that down until we find the gene itself, which is what's going wrong in this particular disease. Inevitably, as soon as we have a gene, a DNA sequence, we can find out what protein that codes for, and we can begin to understand the disease. At the moment, we don't understand anything about the biochemical basis, and the clinical course and the details of that need to be worked out. If we've worked that out, then we should be able to design an effective treatment that's better than the one that exists at the moment.
It's the sheer complexity of the DNA code which makes research so difficult. Everybody is hoping that soon our understanding will improve, and then we will be able to begin to eliminate some of the dreadful genetic diseases that affect so many families. But in the meantime, we will no doubt go on mixing genes in the old-fashioned ways. And no doubt a few couples will be on the losing side in the game of chance. And that's why scientists want to continue research to try to find some of the answers.
And those who are supporting the Powell Bill are saying that they would rather see parents like ourselves and many other hundreds of parents through Britain and other parts of the world for that matter, to go through with the sort of difficulties we've faced, and children like Alan and Geraint to live for only a limited period of time, and to be faced with the suffering that they suffered, and to die, in order to salve the consciences of those who haven't had to face it themselves. And we believe that that is a very selfish point of view to be taken by those who do like to pander to their own consciences and not face up to the reality that the rest of the world has to face to.
What I feel very, very much is that any illness and the suffering, the loss of our boys, will not go in vain. I really feel that everything must be made possible for research to continue in order that more lives aren't lost in this way.
Well, some problems for 10 to 12-year-olds to work on next in Mind Stretchers. That's in 3 minutes.